All right, little lull in the conversation, might as well get started. So I think I mentioned this on Monday, but I am not able to be here for office hours on Friday. So Thursday, I have a lot of availability Thursday if you want to come in and look at your exam from last week. I'm available after lecture today if some of you want to come. So we'll try to find another time. Supposed to get snow on Friday anyway, so like in the afternoon. So if you're not out of town by then, you might want to leave in the morning anyway. All right, so we were talking about things that kill us at the end of last lecture, and how we've made a lot of advances against infectious disease. We just finished a chapter talking about ways to control microbes on surfaces, things like using chemicals, things like using heat, just kind of in general, improving hygiene. So just improving hygiene has just alone dropped our transmission of disease. So now we'll talk about using drugs. That's another big step in fighting infectious disease. Eventually, we'll talk about vaccines as well. We have to kind of understand some things about immunology before that will make sense. So eventually we'll get to that. So thinking about some terminology here before we move forward. And hopefully those of you that want to be pharmacists, hopefully this is especially interesting. But any drug that's considered a chemotherapeutic agent. I mean, when we think about chemotherapy, we think about cancer drugs, but technically, by definition, that's any drug that's used in the treatment of any kind of disease. And then an antimicrobial agent is one that's used for some kind of infection. It could be caused by a protozoa, it could be caused by a fungus, for example. So then if we use the term antibiotic, that's in a strict sense, we would say that that's a drug that's biosynthesized by some kind of organism. And we'll talk about what kind of organisms produce antibiotics. There actually are, the antibiotics that we use though, a lot of them are actually semi-synthetic. We could call those semi-synthetic antibiotics. That's taking the natural product that's produced by an organism, and then we've chemically kind of monkeyed with a side chain or something in order to give that natural product some little better properties, maybe so that it works better. Oh, and I should emphasize here that an antibiotic by definition is going to target bacteria only. Okay, so if you have an antiviral, that targets viruses. If you have an antifungal, that targets fungi and not bacteria. Okay, so again, semi-synthetics, we still call them antibiotics, even though they're not the natural product. There are fully synthetic antibiotics as well. So again, we make those, synthesize them chemically. They're not from organisms, but we still call them antibiotics as long as they, again, are targeting bacteria. So most of our antibiotics in use are either fully natural products or semi-synthetic. There are a few synthetics, like sulfa drugs. All right, so I think it's interesting to do a little history here. Think about kind of how we got to where we are today. The father of chemotherapy, his name was Paul Ehrlich. Make sure I spell this right. Here, whoops, I always have trouble spelling Ehrlich. German. Yeah, Paul Ehrlich. Okay, so this is him right there. He's considered the father of chemotherapy. He was the first one to really think systematically in a way to try to figure out drugs that could actually treat someone once they have an infection. So he was interested in treating syphilis, and he went through a number of arsenic compounds trying to find one that would treat syphilis, and he did find one. I still didn't spell that right. E-H, sorry, let me try this one more time. E-H, Ehrlich, there we go. He tested a number of arsenic compounds. The one that had the best activity against syphilis, he called salvarsan, and that was used for a time. The problem is anything that has arsenic in it, arsenic is pretty toxic to us as well. So he was always searching for what he called the magic bullet. Okay, in combat, a magic bullet would be one that killed your enemy and not your friend. Okay, he was looking for a drug that would kill the pathogen and not be harmful to our cells. Okay, and he had some small success with that. Again, arsenic is not good for us in really any amount. Really, the next person that really found something that had any activity against bacteria was Gerard Domack. He was another German scientist. Europe was way ahead of us in terms of biomedical research back then. Again, this was in the 1930s. And he was working at a big pharmaceutical company, Bayer, or what at that time was, you wouldn't have called it a pharmaceutical company, I don't think, but I think they made aspirin at least back then. And again, he was systematically testing a number of chemicals in a test tube to work against streptococcus, and he found this sulfa drug. Now, at that time, sulfa was used for lots of things, so they had to figure out how to market it so they would make some money. But they had really good, again, activity against streptococcus. So again, a particular bacterial infection, but that saved a lot of lives because streptococcus was the organism, still is the organism that causes scarlet fever. And kind of about that same time, a little bit earlier, actually, is when Alexander Fleming discovered penicillin. About 1929, he was working in his lab in Oxford. So this is him over here. The story goes that he went away on vacation and he left a bunch of his plates, his petri dishes off to the side. He didn't do his dishes, glass petri dishes at the time. And we came back from vacation a couple of weeks later. He saw something that looked like this. He had staphylococcus growing on a plate and this big old mold contaminating it. Well, chance favors the prepared mind, and he noticed that there was this zone around that mold where his staphylococcus weren't growing. So he reasoned that that mold was secreting something that was killing the bacteria. And he thought, this could be useful. And he was able to kind of figure out the active ingredient that was being secreted by that penicillium mold. So he called it penicillin. And he wrote that up in a paper, but he wasn't able to extract it in high enough quantities so that it would be enough to treat an infection in a person. So he kind of gave up on it and went on to researching something else. Well, about 10 years later, Howard Florey was looking for he saw that, this was in the 1930s. He saw that, war was on the horizon and that we really needed a drug to cut down on battlefield infections. And he came upon Fleming's paper, and he decided to assemble a team to try to figure out how to mass produce penicillin. So this is Howard Florey here. Right next to him there is Ernst Chain. Ernst Chain was a organic chemist that helped them. It's basically an organic synthesis that you have to figure out how to, again, grow that mold up in high enough quantities and to extract it. And they were successful in figuring that out. Lots of other people contributed to that as well, but we kind of remember Florey, Chain, and then Fleming because they're the three that got the Nobel Prize for it. Apparently, you can only share 3 people for a Nobel Prize, so they're the ones that are kind of famous for that. So that's kind of an interesting story about how that all happened, and that really spurred the increase in looking at lots of other ways, lots of other potential organisms that could make antibiotics. Selman Waxman, he's famous for really the next big antibiotic that was discovered was streptomycin in 1943. That wasn't in general use until the late 40s though. That's him right here. He was a soil scientist working at Rutgers University. So that's kind of the first American that gained some fame. And streptomycin was such a miracle drug. Penicillin worked against a lot of gram-positives, and kind of narrow in their spectrum of activity. And then streptomycin was the first drug that worked for tuberculosis. So that was a huge scourge at the time. That was just considered a miracle drug when that came out. All right, so that's, and of course, we've been developing antibiotics ever since. Where do antibiotics actually come from? So what kind of organisms make antibiotics? Both bacteria and fungi. Not sure why I have a leftover mark there. There we go. Okay, so mostly soil organisms. I don't know of any examples of an organism that does not primarily live in the soil. There are some other drugs that are isolated from plants. There's a malaria drug that we'll talk about that's made from plants. But as far as I know, any drug that has activity against bacteria either comes from a bacteria or a fungi. So these are the same ones that maybe you talked about in lab this week, but just a couple of examples of bacteria that secretes antibiotics. Bacillus, this one that we've used in the lab a little bit, soil organism. Streptomyces, that's another one that we haven't used in lab, but it's kind of a interesting organism because it grows, this is Streptomyces down here, it grows in these long filaments and it makes reproductive spores. Well, it kind of acts like a fungi, but they're prokaryotic cells. So it's actually a bacteria. And then just some examples of fungi, of course, penicillium, that's this green, it usually has a kind of a greenish color to it, big old fluffy colonies, if you see those growing on some media somewhere. And one that's a little bit less familiar, acremonium. We used to call that genus Cephalosporium, and for some reason they changed the name on it. But we get a whole class of antibiotics. Our cephalosporin antibiotics are secreted by these acremonium molds like that. Okay, so those are just some examples. There's many other genera. All right, so what sort of things do we need to think about when we're using antibiotics, or maybe you're a physician that's prescribing antibiotics? Not just whether the antibiotic will kill the bacteria or not, but there's all sorts of other things to think about, how we administer it and things like that. So we'll go through some of those before we look at the actual drugs. The number one thing that we need to think about whether a drug is going to be in general use or not does it have selective toxicity? So that means, does it target some structure or some pathway, some enzyme that the bacteria has that we don't? Okay, if the drug is not a magic bullet, if it's not only targeting the bacteria, then it's just not going to be safe for us to use. Okay, so we'll write that the drug targets a pathway or structure of bacteria that humans lack. So try to get you to think about that in all the drugs that we talk about. Okay, why does this have selective toxicity? I'll come back to this page in a minute, but here's some mechanisms of action of some examples of antimicrobial drugs. And don't pay attention to the specific drugs here. We're going to study certain ones and not worry about the rest of them. But thinking about by mechanism of action, again, I mean, what does the drug, what structure does the drug target? About half of the drugs that we use now target cell wall synthesis. So again, there's a structure that humans do not have. Animal cells, remember, do not have cell walls. So good selective toxicity. Now, there might be other problems with the drug. It might have some side effects that are not good. It might cause some allergies, but again, and when we're talking about their mechanism, their selective toxicity, targeting something that we don't have. A lot of drugs in use inhibit protein synthesis. So remember, that takes place at the ribosomes, and bacterial ribosomes are slightly different structure. Remember, they're 70S, eukaryotic ribosomes are 80S. So we see a little more side effects with protein synthesis inhibitors, but usually not so much that we can't still use them. Disruption of cytoplasmic membrane. There's not very many there because the cytoplasmic membranes of bacteria are very, very similar to ours. So that's not as big a class of drugs. Some metabolic pathway that we don't have, that bacteria have. We'll talk about that a little bit. A few that inhibit DNA replication and a few that inhibit RNA synthesis. So again, there's some unique enzymes that bacteria have that are a little bit different than the ones that we use. And we will mention a little bit, a few antiviral drugs here, but kind of briefly. So that's an area just that we won't talk about as much, how to inhibit viruses from entering or maybe even leaving a host cell. But okay, so whoops, I have to go back to spectrum of activity here. So selective toxicity, that's that big. We also need to think about, when we're using antibiotics, what kind of bacteria do they target? So remember, they're only targeting bacteria, but do they only target some bacteria, or do they target many different kinds of bacteria? If they're narrow spectrum, that just means that the drug is going to target only specific bacteria. When do we want to use a narrow spectrum antibiotic? If we know what organism we're trying to kill. Okay, oftentimes, say a patient has pneumonia. Oh my gosh, there's a whole bunch of different things that could be causing that pneumonia. An ear infection, all different things. Urinary tract infections, lots of organisms cause those. Strep throat, there's pretty much one. So it might make sense to use an antibiotic that only targets that streptococcus. Sometimes it makes sense to use a broad spectrum antibiotic, one that targets a variety of bacteria. When would you want to use broad spectrum? Obviously, if we don't really know what we're trying to kill, maybe for prophylactic use. What does prophylactic mean? For instance, before my son had his wisdom teeth out, the dentist made him take some penicillin. Okay, so when we use something prophylactically, that means we take it to keep us from getting sick or to keep us from getting an infection. Okay, so before surgery, oftentimes patients will be prescribed some antibiotics to try to cut the chances that they'll get an infection from that surgery. If there's a mixed infection, and by that I just mean if there's numerous species of bacteria that are causing problems at the same time. Or again, you just have no idea, you don't take the time to identify what organism is causing the infection. But there's some risks with taking a broad spectrum antibiotic because we have lots of bacteria in our body already that it's healthy for them to be there. We call it our microbiome. So when we take a broad spectrum antibiotic, that's going to impact our normal microbiota. Okay, and that can have some unintended consequences that may cause what your book refers to as a super infection. A super infection is basically just an infection that happens because of a previous infection or on top of an earlier infection, especially following treatment with broad spectrum antibiotic. Okay, so some examples of common super infections. One is pictured right here. What infection is that when you get this white stuff on your tongue? Nobody's heard of that? Okay, that's called thrush. Okay, so examples of super infections. Thrush, that's caused by a candida fungus, which we probably have, most of us have in low numbers in our mouth anyway. But if we're on some broad spectrum antibiotic that kind of takes out the variety of microbes in our mouth, then the fungus can overgrow. I have a friend that every time she goes on antibiotics for anything, she almost always gets thrush. It's not very common, but and we can take some other drugs to get rid of that. It's not a huge deal. Yeast infections, vaginal yeast infections, again, are more common if you're on antibiotics because it just reduces a normal microbiota vaginally. Another one, C. diff. Okay, we'll talk about that infection more later. We talk specifically about Clostridium. But again, some of your normal organisms in your large intestine, the variety gets kind of knocked down, and any Clostridium difficile that's normally there overgrows, and then the patient can get this really nasty diarrhea. Okay, so again, if we know what organism we're trying to kill, we'd rather just target that organism rather than wipe a bunch of other stuff out. Other things to think about: dosage, routes of administration. We have to consider the toxicity of the drug. So for every drug, we at least we know a range of what we call the therapeutic index. Okay, so this is a way to kind of understand or compare the toxic dose of the drug. Okay, so on top of the equation, or on top of the fraction, the maximum dose of drug a patient can tolerate, the toxic dose. And that might not be the dose that kills a patient. It's the dose at which the side effects are become not acceptable. Okay, so for a cancer drug, a patient might lose their hair. That's a toxic side effect. We consider that safe or acceptable because we're trying to save somebody's life. If you took a drug for an ear infection and it made your hair fall out, that's an unacceptable side effect. So again, the toxic dose is whatever point the side effects become unacceptable, and you compare that to the dose that's necessary or the concentration that's necessary in order to kill the pathogen. Okay, so for example, if you had 100 micrograms was the toxic dose of a drug, and 1 microgram was the dose that was necessary to kill the pathogen, okay, that's a very safe drug. That your therapeutic index is 100. That's probably pretty safe. If it was something like 10 to 1, not so much, because there's differences between individuals and we need to have some, space in there. Okay, so the take home is that the higher the therapeutic index, the safer the drug. All right, so again, tricky thing is that can be, those doses are going to be kind of different in different people. So, okay. Some drugs will kind of stay at concentration in your tissues for longer. So consider the half-life of the drug, the rates at which that half of the drug is eliminated from the blood plasma. So if drugs have a longer half-life, then we don't have to administer them as often. So that's kind of nice. But again, you have to consider the toxic dose. So you don't want to give a whole bunch at once, oftentimes. And there are different administration methods depending on some of the just parameters or qualities of the drug, as well as considering how safe it is. So a lot of drugs can be given orally. That just means by mouth in pill form. A lot of drugs, though, are not able to be given orally, and we'll explain why. So we call them parenteral administration. If this root of this word is enteric or entero, that's Greek for intestinal. Okay, so if it's parenteral, par meaning against or not, okay, not down the intestine, so not able to be swallowed. So we could give them intramuscularly, okay, by injection. That just means injection. We could give them intravenous, by IV, okay, or we could give them topically. So say as a cream, just on the skin. Okay, all those are considered parenteral. All right, so what are the reasons that we wouldn't be able to give a drug orally? Some drugs just are not absorbed well through the small intestine. And the first edition of penicillin that we came out with, that was the case. Okay, it wasn't able to be given orally because patients would mostly just pee it all out. It didn't get absorbed well into our tissues where it needed to be. In addition, some drugs lose their activity in stomach acid. Okay, we call that being acid labile. Okay, that means they degrade in the stomach. And if you have a drug that has a very low therapeutic index, meaning the toxicity is really close to the dose that needs to kill the pathogen, then you're probably not going to want to give it orally, because people make mistakes, right? They forget to take a dose and then maybe they take a couple at once, or they were supposed to take one pill and they take 3 or 4, right? So you need to have some buffer room in terms of how toxic it is if you're going to administer it orally. Okay, our drugs with really low therapeutic index, we're always going to give by IV, just so we can dial it in exactly. All right, so in terms of, you know, the concentration of the drug, maybe in our tissues, if we administer it by IV, we can get that concentration up really high. And again, we can kind of dial that into whatever we want. We can keep that up high if we want. Usually there's, pictures like this, I see that the injection is maybe spiked up a little bit higher. They didn't draw it that way, that's fine. Orally, again, when it's absorbed into the bloodstream, that typically it's not going to be as high as IV anyway. And of course, topically is probably going to be lower yet. Okay. Drug interactions. Whenever you go to the doctor, you have some procedure, they always want to know what drugs are you taking. They always want to make sure that one drug is not going to interfere with the action of another drug. But actually, sometimes drugs can act synergistically. So sometimes, you know, we'll give several antibiotics together because the action of one antibiotic actually improves the efficiency of another. So we'll see some examples of that, how that can benefit the patient. But sometimes, again, one drug will keep another drug from working. You wouldn't want to use an antibiotic that kept bacteria from growing with an antibiotic that only worked on growing bacteria. Okay, because they would sort of, you know, the other one wouldn't work. Some antibiotics will have an effect if you're taking birth control medication and make that not work. So have to kind of pay attention and know how drugs work and make sure that they're going to be beneficial to the patient. And lastly, you have to think about whether the organism that you're trying to kill is susceptible. So there could be reasons that it's not susceptible. Maybe you're trying to kill a bacteria that doesn't have a cell wall with penicillin. That's just not going to work. Okay. We might be able to, but the other reason is the organism may have acquired some kind of resistance. So we can test that by doing a Kirby-Bauer test. Okay, so unfortunately, because of the ice day last week, we didn't get to do that in lab, but we certainly talked about it. If you're not in lab, or if you want to brush up for your exam yet to come, a Kirby-Bauer test, you make a lawn of bacteria on a special kind of media, Mueller-Hinton agar, and then you put antibiotic discs that are impregnated with, in this case, they did a bunch of different antibiotics, not just 4 like we would have done in class. And then you incubate and you look how that disc affects the growth of the bacteria. So this antibiotic right there has a pretty big zone around it. So it's likely that that organism is susceptible to that antibiotic. The antibiotic is keeping it from growing. This one right here, the organism's growing right up to the disc, so we would say that organism is resistant. Okay, so we have charts that take into account the diffusion rate of antibiotics, and we have, you know, different size zones to have it considered susceptible or resistant. In general, the bigger the zone, the more likely the organism is susceptible to that antibiotic. All right, so here's a picture from your book. This is very similar to the one that I showed you before that was from our previous textbook, but that just kind of a roundup of some examples of different antibiotics and how they work on a bacteria cell. So again, don't get wrapped up in all those drugs. We're not going to do that many. We are going to talk about some in particular. Okay, so let's see. Yeah, okay. I have a couple questions that I'll ask, but they're coming up soon. Okay. So, thinking about mechanisms of action of different drugs. I mentioned that about half of the drugs that we use target cell walls. So let's start with that. There's a huge class of antibiotics that inhibit cell wall synthesis, and those are called the beta-lactam antibiotics. Which penicillin is one. Okay, so what they all have in common, I'll be showing you lots of structures, not because you have to memorize and draw out these structures, but you should notice things about them and sort of be able to maybe identify parts of those structures. So for example, all of the beta-lactam antibiotics, here's some examples here, they all have this beta-lactam ring. Okay, so they highlight that in green here. It's actually, if you're into the chemistry of it, a lactam is a cyclic amide, and the nitrogen here is the beta. Sorry, the nitrogen atom is attached to the beta carbon. So the beta carbon is like the carbon right next to the nitrogen in this case. Sorry, yeah, so if this is the carbonyl carbon right there, the nitrogen is the one right next to it. So anyway, that's where the name beta-lactam comes from. We have lots of different beta-lactam drugs. Penicillin is one. Methicillin, that's another one we're going to mention kind of briefly. The carbapenems, we're not going to go into those, but when you hear about kind of super bugs, ones that are resistant to lots of things, sometimes we talk about carbapenem antibiotics there. Cephalosporins, it's another huge class of beta-lactam antibiotics. Okay, all the beta-lactams have the same mechanism of action. So let's look at that a little bit more closely. Thinking about cell wall structure. Okay, so thinking back to something we talked about a month ago, we talked about how the cell wall grows. And we talked about a couple of enzymes that are important for that peptidoglycan structure to grow. Autolysins, remember the autolysins are what cuts the bond between the two different kinds of sugars that make up peptidoglycan, as well as cutting those amino acid cross bridges between the layers of the sugars. And then there are certain enzymes that reforms those sugar linkages once the new sugars are brought in. The other enzyme that we talked about are the transpeptidases. And the transpeptidases are the ones, enzymes that reform those amino acid cross bridges. Okay, so again, these are amino acid cross linkages. So in this picture here, these, I don't know, those are purple or blue or whatever, those are other amino acid bridges. So normally the new sugars get cut, the new sugars are brought in. So if you look really closely here, they're outlined in white. If you go across this way, so normally we get these new sugars, and the transpeptidases, reform these amino acid bridges. That's what's supposed to happen. But if the bacteria cell is in the presence of some antibiotic, so all these little green specks here, that's supposed to be some beta-lactam antibiotic. These green cross bridges that should be forming are not. Okay, the antibiotic inhibits the transpeptidase enzyme from making those cross bridges. And so what you end up with over time as the bacteria cell is trying to grow and expand its cell wall is you get peptidoglycan that's not sound. Okay, and because that cell wall is not totally intact, it's going to be really susceptible to osmotic pressures. So in other words, when the cells are normally in a hypotonic environment, that water is trying to enter into the cell. And eventually, again, if the cell wall is not intact, then that cell wall will not stay intact and it will burst. Okay. So it's not really the beta lactam itself, it's because the cell wall weakens and then it's susceptible to water coming in and lysing the cell. All right, so looking at that in a little bit more detail, again, if you want to impress your chemistry professors, here's the structure of penicillin. Okay, so again, don't recognize it. I mean, don't memorize it. Here's the beta lactam ring. Here is the structure of usually a couple of amino acids that are almost always present in those amino acid cross bridges. Okay, so D-ala, D-ala, D-alanine, D-alanine. Amino acids are present in two different isomers. There's kind of a left-handed isomer and a right-handed isomer. And proteins always have the other isomer, the L isomer, and we don't find the D-alanine in any protein, but it is found in peptidoglycan of bacteria. So that's kind of a unique place that we find at. And if you look at the structure there of D-ala, D-ala, and the structure of penicillin, and you superimpose the two, they fit right on top of each other. So actually, the penicillin binds where the two amino acids, alanines, typically bind. And that's the transpeptidase. So I tried to write this out here. Penicillin mimics those amino acids. So basically, penicillin binds to the active site of transpeptidase. Okay, anything that binds to the active site of an enzyme, what do we call that? What kind of inhibitor? A competitive inhibitor. So we'll say it's like a competitive inhibitor. Penicillin is like a competitive inhibitor. It binds to the active site of transpeptidase. And then you don't get the right product. You don't get that's supposed to be there. Now, I say like a competitive inhibitor because we don't actually call it a competitive inhibitor because it's permanent. Usually inhibitors are kind of transiently binding. So it's like a competitive inhibitor, but it's permanent. So again, understand that if a bacteria cell is not actively growing, it's not inserting new sugar subunits into a peptidoglycan to expand its cell wall, it's not going to be susceptible to penicillin. If it's just kind of sitting out, hanging out there, penicillin is not going to have a lot of effect. So I think I mentioned that before, in the log phase of growth of any bacteria, it's going to be, those organisms are more susceptible to antibiotics. Okay. All right, so let's try a couple of practice questions here before we move on. We just talked about this, but let's test yourself. Try not to look back at your notes. So by the way, I was going to make fun of myself a little bit because I actually came to class at 12:30 and nobody was here. I was like, gosh, everybody left early for spring break. I can't believe it. And then I realized, cancel time. Back to my office. I'm glad more of you showed up than what we're here at 12:30. Okay, so a low therapeutic index. That means that the toxic dose is really close to the therapeutic dose. So C, we want to really carefully dose this so we don't exceed the toxic concentration. I can see that you might have mixed it up with B here. The rest here, that doesn't really have anything to do with it. So, I don't know, that might be right in a weird situation. But sometimes it's confusing. Oh, wait, do we want it high or do we want it low? So that can be the confusing thing. And do you remember our history people here? See some heads shaking. Okay, so those things you got to study a little bit. How many people say A? Yeah, okay, good. Yeah, that's the one. All right, good. I know clicker questions are more fun, but so yeah, so Fleming was the original discovered penicillin. Domock was the sulfa drugs. We're going to talk more about sulfa drugs. Ehrlich was the magic bullets, the father of chemotherapy. Florian Chain mass-produced penicillin. Okay. All right, good. So before we jump into our specific drugs, our little kind of mini pharmacology course here, I'm going to throw a lot of information at you. The number one thing to remember about the drugs is how they work. Okay, so I'm going to emphasize that. I'm going to talk about other things like what's the spectrum of activity and how do we administer it, mostly because I want you to get familiar with that terminology. If I were to ask you something about a drug on an exam, I would give you more information like this is a drug that does this, this, and this, and it does this, not just this drug is broad spectrum or whatever. Okay, so kind of focus on the mechanism of action, focus on the advantages, like, oh, why would we want to use this drug? What are some infections that it works really well against? What's really nice about it? And also disadvantages. So what kind of side effects do we have to deal with if we're going to use this drug? Okay. All right, so I hope you find it interesting. We've all been on antibiotics at some point in our life, and we'll also get to talking about resistance and problems with antibiotics a little bit too. Let's start with our narrow spectrum penicillins. So penicillin G, that was the first, that was a natural product made by that Penicillium, I think it was Chrysogenum was the species of that mold. Why G? Well, when they were trying to, mass produce penicillin, they had lots of different ways that they were doing this organic synthesis. And so they had kind of different variations of penicillin. So the, penicillin A, penicillin B, penicillin C, and the G one was the one that showed the best activity against certain, gram-positive microbes. So that's where the G comes from. All right, so again, effective against gram-positives. Again, this is the first one that we came out with in 1941 or whatever. It's cheap. Flory and Chain actually argued about whether to patent the drug or not, and they ended up not, which is totally unheard of today. Imagine Pfizer saying, "Oh, we're not gonna patent Paxlovid. We're just gonna let everybody use it." For one, they invested millions of dollars in producing it, so that's why they don't do it anymore. But that was great at the time. It made it immediately available. Lots of different companies could make it right away, and it was very cheap. And again, saved lots and lots of lives. That penicillin penetrates pretty well when you administer it a certain way. Had very low side effects for a drug. It did require injection. Okay, so next time you see your grandparents, maybe you'll see them over spring break. Ask them if they remember what they remember about taking penicillin when they were a kid. My father, who was a young boy in the 40s, He remembers Doc Martin coming to his house when he was sick, and he had to get an injection of penicillin in his rear end. Okay, my generation, when I took penicillin, I can remember drinking it, and it just tasted like the worst thing that you can imagine. I had to eat a cracker or something to try to get that taste out of your mouth. Your generation, what do you remember? You remember taking this bubblegum flavored stuff, right? Like, either way you look at it, either, we're improving or we're getting soft. I don't know. Okay. But it had to be injected back then because that original form of penicillin was degraded by the stomach acid. So again, we say acid label. And it was absorbed poorly through the small intestine. So for those reasons, they had to be given by injection. Penicillin now, it's penicillin V. We can take that orally. We don't use penicillin very much anymore because there's so much resistance to it. But what's different, penicillin V, actually, they found that if they grew the mold on a slightly different media, the secreted product by the mold was slightly different, and it was absorbed. I still don't think it's absorbed very well. I think you have to take a lot of it, but it withstands the stomach acid. Now, another problem, the main problem we have with penicillin is most organisms are resistant to it now. And actually, Alexander Fleming predicted that that was going to happen, even right after we started making it. Why is it resistant? Well, a lot of organisms can make enzymes called beta-lactamases that will destroy the activity of the drug. So let's talk about that before we go any further. We'll talk about this more later. So beta-lactamase, again, sometimes it's easy to get mixed up. Wait, who's doing what? The bacteria produce this enzyme, beta-lactamase. Okay, and it acts on, remember, here's the penicillin. It acts on the penicillin and breaks this bond right here in that beta-lactam ring. So you see how that opens the ring up a little bit, and just that one little change makes it not fit into the active site of transpeptidases. Okay, so these are enzymes that some bacteria acquire the ability to make that makes it so they can survive in the presence of a beta-lactam antibiotic like penicillin. What was I just going to say? So how do they acquire the ability to make this enzyme? It's usually not just a little point mutation or two that would suddenly enable them to make a whole new protein. Usually they acquire this by some horizontal gene transfer, like conjugation, getting a plasmid from another organism. Okay. So we'll talk more about resistance a little bit later, the different mechanisms of how that happens. Kind of a second generation penicillin. Methicillin came out, I believe, in 1951, 1950s. And we don't use methicillin anymore. But I did want to mention it because it's kind of one of the early semi-synthetic derivatives of penicillin, meaning you're just kind of, again, monkeying with the side chain a little bit. And they did that because it had a little bulkier side chain, and they thought that that was going to protect the drug from beta-lactamases produced by bacteria. And it did for a while. A little more side effects than penicillin, some kidney toxicity. That's kind of one of the more common problems with taking antibiotics. It was acid labile, so again, that means that it had to be injected. But the main reason that I bring it up is because we have this infection now, and this showed up soon after methicillin came out, and that's why they named it MRSA. Okay, that stands for methicillin-resistant Staph aureus. And so this is a strain of Staphylococcus aureus that's not only resistant to methicillin. If it was only resistant to that, it wouldn't be a problem anymore. but it's resistant to all the beta-lactam antibiotics and lots of other ones that are commonly used as well. Okay, so often presents as a skin infection, but it can, infect, when somebody has surgery and then it's a real problem. Not particularly aggressive strain of Staph aureus, but just hard to kill. So can cause a lot of problems. Okay, and while we're on the topic of problems, I thought good time to talk about penicillin allergies. Okay, so hypersensitivities to penicillin depends on what source you look at, how many people are allergic to penicillin. I've heard as many as 10%. The number is probably more like 1 to 2%. I know that, for me, my whole life I've been told, oh, you're allergic to penicillin because I had a little rash when I was 5. So am I still? Maybe not. But if somebody does show, a little rash, or maybe they get some hives while they're taking penicillin, typically the doctor will say, okay, stop taking it. We'll switch to a different drug. Because maybe the next time you take penicillin, you might have more of a hypersensitivity. If you think most people, like the first time they get poison ivy, it's because they become allergic to that oil and poison ivy. Like the first time I had it, I just had a little bit. The next time I had it, I got a lot worse. And so sometimes they're kind of additive. And maybe you have a rash the first time, maybe the next time you have penicillin, you can actually go into shock. About 200 people die in the US every year just from taking penicillin. So sometimes people actually develop, or they produce antibodies to the metabolized penicillin, and that can kind of set off a reaction that actually causes some people to die. So if you've ever been told you're allergic to penicillin, no doctor will ever prescribe penicillin for you. Although today, with they are starting more and more, if you go to an allergist, they will test you to see if you actually are allergic to it. So that's probably a good idea. Taking, some kind of beta-lactam is much cheaper than a lot of other drugs. And again, if you're allergic to penicillin, you're allergic to any beta-lactam drug. So methicillin or more that we'll talk about here. And you might think, again, having a little rash or maybe some hives is not a problem. Dr. Baxter, who some of you know maybe from lab, when he had penicillin as a child, he had hives for 9 months. That's what he claims. So you can imagine having hives for 9 months. Like, oh, I can't even imagine. Hopefully Benadryl worked. But okay, so usually just a little rash, but it has the potential to be a lot more serious than that. Even things like fever or diarrhea, like, well, is that the penicillin or is that something else? Kind of hard to tell. All right, so again, we started kind of messing with that natural product of penicillin and started monkeying with some side chains, and we were able to produce some more broad spectrum beta-lactam. So amoxicillin, we came out with that in 1972, I believe. And I'm thinking, okay, so this is a drug, right? That's the bubble gum flavor that we take for a lot of childhood illnesses, things like ear infections, strep throat. Whoops. And I was kind of, I cannot, let's try again. Strep throat. I was kind of surprised that that came out in 1972. Maybe they didn't start adding the bubblegum flavor till later, or maybe that just didn't make its way to rural Ohio when I was a child in the 70s. But again, still mad that I missed out on that. I had strep throat all the time, so I can remember taking drugs all the time. So again, nice thing about this, acid stable. We can take it by mouth. Good absorption. We don't have to dose it as frequently. I think it's 3 times a day, which seems kind of often if you're trying to give it to a kid, but must be less often than what penicillin had to be. Still used today, but resistance is a growing problem. So to try to extend the use of amoxicillin, typically it's marketed today as something called augmentin. Okay, if you augment something, that just means you add to it to make it better. So we've added something to the amoxicillin, clavulanic acid. This is a beta lactamase inhibitor. It's actually another antibiotic, but it must bind to amoxicillin to kind of protect that beta lactam ring so that enzymes, again, produced by bacteria, beta lactamases, can't harm that beta lactam ring. Okay, so that's, again, when we market those two things together, okay, they work synergistically. Those two drugs help each other, and it's an effort to make amoxicillin useful for a longer period of time to kind of hold off those resistance problems. Other beta lactams. Cephalosporins are in wide use, and there are many, many different cephalosporins. They have actually 2 side chains that are variable that we've monkeyed around with a little bit. They're produced by the fungi Acremonium cephalosporium. That's why we call them cephalosporins. And all the cephalosporins start with that prefix ceph. Okay, so cephalexin is an example. Ceftriaxone is another one. So again, you see that's CEPH, and that's a cephalosporin. Most drugs have a trade name in addition to what the drug is. So Keflex, that may be more recognizable, I don't know, but that's the trade name for cephalexin. Okay, so yeah, if you want to be a pharmacist, know what you're getting into. There's all sorts of different names for the same thing. And these drugs, again, they're beta lactam, so same mechanism of action. They bind to transpeptidases and eventually the cell wall lyses. Not as much trouble with resistance yet. There are all sorts of generations. Okay, so the first cephalosporins that we produced, those are the first generation. We go all the way down to 5th generation cephalosporins, the more recent ones. The early generation ones were very safe, but they were very narrow spectrum. And then over the generations, they've become more broad spectrum, but also the therapeutic index has shrunk. Okay, so some of the later generation ones, like the higher generations ones, they actually will cross the blood-brain barrier. Most pathogens can't do that, but the ones that cause meningitis can. So we need drugs that will follow them there. So they can cross that blood-brain barrier, but again, there's toxicity issues. So we have to dose that out just right and only give them by IV. And then the 5th generation, that's one of the drugs. It's kind of different enough from beta lactams that it sometimes will show some activity against MRSA. And I've heard that even some people that are allergic to penicillin are able to take the later generation cephalosporins as well. All right, so still on cell wall inhibitors, there's a few that inhibit the cell wall in a different way. They're not beta lactams, so they don't, again, bind to transpeptidases. They work in a little different way. Vancomycin. So here's a structure of vancomycin. Just this huge structure. It's actually a physical block. So it binds to that peptidoglycan and keeps those peptide bridges from forming. It's narrow spectrum, so it only works on gram positives. It's kind of best known for usually being the drug of choice to treat MRSA. So it does work there. The other thing that it's commonly used for is C. diff. So most of the time it has to be administered by injection because it's not absorbed very well. But if you're treating C. diff, that's the one exception. You can give it orally, and then it kind of gets to the place where it needs to work, our GI tract. Typically, when we first came out with vancomycin, I think this has been around since the 50s. When we first started to synthesize it, we had a lot of trouble with patients having like kidney problems after they took it. Since then, we've gotten better at synthesizing it apparently and got rid of impurities, so it's a little bit safer to take, but it's always been considered sort of, you try other drugs first, and if they don't work, then you try vancomycin. Now there are strains of bacteria that have been found that are even resistant to vancomycin. So it starts to be kind of an alphabet soup here, but VRE, that stands for vancomycin resistant enterococci. That's a medical emergency, because if we have a strain of bacteria that's even resistant to vancomycin, we don't have a lot left to treat it. So that's one of those kind of super bugs that you might have heard about. Typically, where would you pick up superbugs? Typically, if you're somewhere where antibiotics are used a lot, like in a hospital. Okay, so people that are otherwise healthy probably are not going to have a problem with that. Bacitracin is another cell wall inhibitor. Works, again, a little different mechanism, does work on peptidoglycan. It keeps those new sugar subunits, NAM and NAG, that are synthesized inside of the cell, keeps them from passing through the cell membrane to get to the cell wall. So it's kind of a physical block in that channel. Also, it's narrow spectrum, only targeting gram positives. Again, not a whole, it's kind of this big complicated structure. This is a drug that we don't take orally even. We don't inject it either because it has high toxicity to the kidneys. So again, we use this topically, and don't confuse. There's another drug called Bactrim that's different. So bacitracin, cell wall inhibitor. Bactrim is actually a sulfa drug. We'll talk about that one later. Anybody, you probably all use bacitracin like lots of times topically. This is one of the antibiotics in triple antibiotic ointment. So some of you are saying, what are you talking about? I've never used that before. Great, if you get a cut on your skin, it's a really good idea to not only wash it out really good, but put some antibiotics on there to keep it from getting infected. Okay, so that's one of them. The two other ones we'll get to, but they work a different mechanism. So we kind of, try to kill bacteria several different ways. Okay. All right. One more cell wall inhibitor. I'm probably making your head spin a little bit. Isoniazid. Kind of fun to say, isoniazid. This is one of our antibiotics that's fully synthetic. It acts to prevent the synthesis of mycolic acid. So I think actually OpenStax calls this isoniazid as the mechanism is preventing a biosynthetic pathway. But because it's a pathway to make something that's in the cell wall, you could consider it a cell wall inhibitor. Really, both would be correct. Where do we find mycolic acid? What kind of bacteria have mycolic acid? Yes, acid fast. So this is really narrow spectrum. Knowing the mechanism of action, you understand that it's narrow spectrum. This is going to inhibit the Mycobacterium. Remember, that's, yes, the acid fast. Okay, so we're going to use this on things like TB and leprosy. One of the drawbacks here is it causes a lot of nausea. Okay, so it's hard to keep taking a drug, even when you're trying to treat a bad infection, it's hard to keep taking it when it makes you feel lousy. Okay, and we talked about streptomycin, Solomon Waxman making streptomycin the first drug that was effective against TB. Whenever we treat TB, we'll talk about this more later, we usually treat it with lots of different antibiotics, at least 4 different ones, because it just takes a long time to kill. They grow really slowly and they die really slowly too. So it's a problem with treating TB. The drugs that used to treat it, they make you feel lousy and they make you feel tired. So that's a problem. Okay, so let me give you a practice question, and then I want to see if you've been kind of listening to what we just talked about, or try not to look at your notes. Which of those is not a beta-lactam? Oh, I did this too quickly. I spelled it wrong. I spelled one of them wrong. Okay, that should be an I there, amoxicillin. Okay, the one that's not a beta-lactam is the bacitracin. All right, so you may be thinking, how am I going to keep all this straight? Okay, this is about half of the drugs. We're about halfway there. What I like to do, I've drawn up a partial concept map here, and this is in your notes. I've partially filled it out with the ones that we've got so far. When I was first teaching this class and thinking about what drugs to use, and I had to learn it myself, I drew this out over and over and over again. I think this is better than flashcards. Some of you like to do flashcards. So here I've got the cell wall inhibitors. These 4 are the beta-lactams. There's many more that we didn't talk about. These are just the ones that I've chosen. Other drugs that are beta-lactam, they're cell wall inhibitors, they're just not beta-lactams. Okay, so there's really 4 other mechanisms that we're going to talk about. We're going to have to talk about examples of drugs in each one of those. So fill this out, at some point once we get done talking about all the other drugs. And again, this might help you remember the mechanisms and kind of think about what kind of drug they are. Okay. So again, I think that's a good place to stop. We're going to start next with the protein synthesis inhibitors. That's kind of the second big class. Have a good break. Maybe do a little homework. There's chapter 12 that you can be working on, chapter 6, the viruses. Have a good break.